session, the one and only. My name is Clarissa, and I'm going to talk about diversification islands within the continent, bringing to you a case study in Antilophia manakins. So, diversification of species can happen through several processes, and one of them is through genetic bottlenecks, via founder effects, or as a result of drastic population decline. And in this context, islands can be seen as great models because of the colonization events and associated bottlenecks that lead to different biological entities. Islands tend to have reduced genetic diversity compared to mainland ancestors because they are a little sample of the genetic diversity of the continental populations, which is further accentuated by genetic drift and inbreeding because of the loss of the gene flow with the continental populations. These processes, along with local adaptation, can culminate in reproductive isolation and speciation. So here I brought to you a case study that is led by my supervisor, where only 200 meters of sea separates populations of birds, leading to strong genetic structure in a bird population. But many cases analogous to this might be happening in other organisms with different dispersal abilities. This is a classic example of islands and where there is a little piece of land that is isolated by the ocean. However, there might be analogous examples in the land. For example, here we have Mount Roraima in South America that is surrounded by open grasslands. However, if you see in the highland, you will get a much more forested landscape. So populations who live there, they might be isolated from their ancestral populations. Therefore, because they are surrounded by their known preferred habitats. So this works analogous to an island in the middle of the ocean. So while elevation can create islands within the continent, and because of that, they could be centers of endemism. In South America, for example, we have the Andes that have elevation from 4,000 to 7,000 meters. And needless to say that the Andes, they host a lot of endemic species because of the isolation process, but also because of the processes that led for them to adapt to the high elevation. However, there are multiple little islands in the rest of South America that are less studied, even though they also host lots of endemic species. So lots of little mountains that have 1,000 meters and sometimes even lower than that host many endemic species. But because they are understudied here, we propose to investigate the historical processes that could lead to diversification in those little islands within the continent. So here I introduced the model that we're using, those two sister species of manakins, Antilophia galliata and Antilophia bokermani. They are nested within a very charismatic group of manakin birds, those blue little guys. They are famous for their happy lacking system, where there are lots of males jumping one on top of the other, but only one of them mates. In our systems, though, they do not have such elaborate legs. They tend to have some display that flying back and forth, but they tend to be much more territorial than having those crazy leg displays. And one of the hypotheses for this is that they live in the Brazilian savanna, which might offer harsher conditions compared to the rainforest where the other blue guys are found. So they cannot waste resources having lots of males jumping one on top of the other and one of them mating. So these guys, they tend to be found more closer to rivers where the tree stratum is a little bit taller. But even though they are closer to river, they are still in the middle of the Brazilian savanna. So they are exposed to the high temperatures and the high seasonality that the savannas offer. And this is its sister species, Antilophia bokermani. It's found in a much more restricted distribution in this little green point in the middle of a semi-arid sea. So this could be an island that offers it conditions to thrive in the middle of a very semi-desert, harsher conditions. And because of this restricted distribution, it is a critically endangered bird. This is how it looks like seen from above, and this is how it looks like through the bird's perspective, so to say. So it is a little oasis in the middle of the semi-arid environment. So the objectives of this work were to estimate the demographical history of this species, then compare the contemporary genetic diversity to know how these species differ, And finally, to characterize the genomic regions of accentuated differentiation, to point out what could be some potential candidates that explain the phenotypic differences among these species. And to this end, we used museum samples from Antilope galeata from the north of the distribution, that is the closest possible to the Antilope bokermani population, but also some populations from the south where there is this ecotone with the rainforest below. For the Antilope bokermani, we counted on a collaboration of our colleagues that work directly on preserving this species of birds. They do a great job in managing their numbers, such as creating those little creeks to expand their habitat and give them more conditions to strive. So we used those tissue samples to generate whole genome resequencing, and with coverage around 10x, maximum 15x, and we aligned them with its closely related species, Chiroschifia lanceolata, retrieving the great majority of the reeds. So what we found is, as a principal component analysis, they strongly differ, which is further supported by admixture analysis. You see that they clearly are split compared to other models with K above 2. So we get this K equals 2 to estimate the demographic history of this species. And what this analysis shows is that they might have diverged, like the split happened across 110,000 years ago. And what it says is that bokermani has steadily reduced its population size until what we have today, while galeata has maintained its ancestral effective population size. There are some arrows there that suggest that there is some ancestral introgression, but that could also mean that there hasn't been enough time for them to completely split their differences. We further ran some PSMC analysis to understand a little bit more on how this historical split happened. And it's consistent with our GADMAT analysis, as it shows a steady decreasing in the bokermani effective population size, from the moment of the split where they had 200,000 individuals, decreasing to what the most contemporary value would be 4,000 individuals. For galeata, we have more or less the same effective population size, but this analysis reveals a massive increase in their numbers, and then from the last interglacial until the last glacial maximum, followed by a considerable decline. One of the explanations for this is due to the crazy changes that happened during the last glacial maximum. It became much drier during this period, which allowed the expansion of the vegetation that are favored in drier conditions. It's not very easy to read here, but believe me, there are a lot of those habitats there that are composed by open grasslands, savannas, and steppes. So these might have favored the expansion of galeata as long as there were some rivers with a higher tree stratum. And as long as it was expanding, there was a semi-arid at the corner of Brazil that also expanded. So as the arid habitat has increased, the galeata escaped and leaving that little island of bokermani on the top of it. And why did the decrease happen? Because later, after the last glacial maximum, the rainforest expanded, such as the Amazon or the Atlantic forest. That's why we could explain this decrease in the numbers. Considering their genetic diversity, we found that Antilope bokermani had around 7% of heterozygosity, which could be considered not that low, because the census is like a 213 individuals maximum. So this is not too low for such little numbers, and it's not that low compared to other endangered species. When we get this and see in the whole genome, we have some patterns that we can point to. First one is that heterozygosity tends to be higher at the ends of the chromosome, possibly because of the high repeat content, especially compared to galeata, that's also a pattern there. And something to pay attention is that the Z chromosome, the sex chromosome, is considerably depleted in heterozygosity. In other words, there is very high homozygosity in both species, which I will return to later. Finally, there are many regions in the chromosomes that are also depleted in heterozygosity. In other words, there might be some runs of homozygosity that are happening in this critically endangered species. When we give a little zoom on chromosome 1, this can be highlighted. And in red, there are regions that are above 1 mega base pairs. They can vary among regions, but there are some that are consistently high in homozygosity. And these regions, they mean that the both parents have exactly the same sequence, so no mutation or recombination. They are exactly the same, and that reveals high relatedness and high rates of inbreeding. Here is an equivalent in Galeata for a comparison. And we found that 16.2% of the genome in Bocchermani corresponds to these high runs of homozygosity, so suggestive of high rates of inbreeding and of ancient bottleneck effects. Finally, we characterize the genomic regions of accentuated differentiation using fixation index. And among these two species, even though they diverged 100,000 years ago, the FST is considerably high, 0.15, which is possibly because of this large drift in isolation. Here I highlighted the most accentuated differentiation regions using hidden Markov models, and they were mostly enriched in geoterms for ion channel activity, mostly active. And this combination of geoterms in the literature have been associated with neuronal activity and neuronal development. I'm not really sure what this means, but geoterms are always confusing. I'm going to dig down it later to see what this could imply. Finally, remember that I said that the Z chromosome was very high in homozygosity. Well, it seems like they were high, but for different variants, to the point that the FST in the Z chromosome is higher than the rest of the chromosome. In other words, it's possible that divergence among these two species was more accelerated in the Z chromosome than for the rest of the autosomes. If we select the most outlier regions, like the 0.1% of the most differentiated regions, guess what? We find color that some candidates for colors, some candidates for melanosome development and production of melanin that could explain the differences between these two males. So in summary, we found that divergence occurred during big climatic shifts and that ancient bottleneck effects in contemporary breeding has led to the diversification of these species on the top of a little island in the middle of the continent. And this led to highly differentiated genomic regions with some phenotypic effects, including the color genes. And finally, I would like to highlight that these islands within the continent, they are not so tall, but they have very particular history with a great potential to lead to different species and endemism. And with that, I'd like to thank all my collaborators, and I'm happy to take any questions if we have time. The next step is characterizing those runs of homozygosity because we're not really sure what is going on there. So what are those that are consistently homozygous? Is there any clue about what's going on there? Yes?
So what's the current conservation status of Bocchermani? Is that a protected area? Can you talk a little bit about that?
Okay, so currently there is an NGO that is looking after those birds. There are 213 adults at the moment, but increasing in numbers. So they are doing some management plans to increase their habitat. Well, thank you.
Okay, just the green button.
We go here.
Some downloaded here.
Yeah, it go here. It's
Yes.
that one.
Yeah.
And present.
Present. Yes, exactly.
It's on.
Contact zones are speciation laboratories, and they can arise by basically two mechanisms, in a very simple way. By primary contact zone, when we have an original ancestral population, that is experiencing an environmental gradient, okay? And what we expect is to get local adaptation to that environmental gradient, but we're gonna get a hybrid zone between the individuals that are adapting to one side of the gradient and individuals that are adapting to the other side of the gradient. They can also rise by secondary contact zone, where we have an original population that is split by a barrier. Okay? Some time passes, probably drift, a little bit of even natural selection and even sexual selection, and then when that barrier disappears for some reason, we get those populations together again. And if they haven't differed enough, okay, they get to hybridize. The problem with this system is that you have two very different mechanisms, but the same pattern. And this is usually what we see in the present, right? So we have to tease apart what are the reasons for one or the other process. Okay. So before genetics, we will go and look for that environmental gradient, okay, and see how phenotypic differences would match to that environmental gradient. Now with genetics, we can test models of diversification. We can test a model in which we have an ancestral population that divides with gene flow, okay? And we can have a model in which divergence can happen, and then we get gene flow when those populations have been separated for some time. Our study organism is a hummingbird, and I don't know if many of you know, but Ecuador is a tiny, tiny country. So finding a species that has almost its whole range in that little country is a given. It's a given. It's something unique. And that's one of the reasons we started working with this bird. But it also has other aspects that are very interesting. The species has two subspecies. And they live in the high Andes. We have the purple-hooded ones, and they live in most of the distribution, okay? And we have the green gorgeted ones that live in the middle of the distribution. And I'm gonna be talking about these two subspecies over and over during the talk. The females are boring, they are all the same, so they are not very informative when you are trying to understand evolution behind these plumage patterns. They are also very interesting because they live in these paramo areas. These are neotropical highlands that are much more diverse than temperate highlands. And this is a model from actual palynological data from the paramos of Colombia. And what we see is that during the last glacial maximum, the paramos in brown, they are connected, but in the present, some of them disappear, some of them get separated. So it's a very cool system to think about demographics and biogeography in general. We started asking two questions: if these two subspecies were independent evolutionary lineages, and if these paramo islands were actually getting connected and disconnected, and actually disconnected in the present. We started in the golden era of mitochondrial DNA. Of course, we found squad, and we decided to design our own microsatellite primers, and we got 10 microsatellites. And this is where the story started to get interesting. What we have here is a north to south pattern of divergence, right? We have the northern population that is mostly a group. This is the best K. Then we have the central population, Chimborazo, in green. And then we have the southern population in Sangay. What we are seeing is a little bit of a mixture in between north and center, and a little bit of a mixture, sometimes a lot, between the center and the south. Okay? What we realized is that we were picking on this probably northern contact zone. There is actually a picture of a Chimborazo living in very close to Apagua. That is the red star in the map. Okay, so that we were okay with that. But we realized that we weren't sampling a potential contact zone south of Chimborazo. So all this area wasn't sampled. We had to go back to the field and see what was going on. And we also compared the morphometry of these individuals. the different populations, they are very similar, okay? There are not many individuals from the south because those are the ones that we collected. They weren't, these specimens, they weren't in any collections, but they are very similar, right? In between the three populations. So we decided to go genomic, and from some of our countries in the south, global south, is not so easy. It takes some time and some money for us. We asked the same two questions, but then we wanted to see if there was actually a southern contact zone, and we wanted to know what genes determine that change in plumage. And there is something that I forgot to tell you that is really cool, which is that this individual that is genetically admixed, like 50-50, is a male. And he looks like the male, how males have to look for Jamesoni, which means that we have a genetically admixed male that looks like a full male of that subspecies, okay? Not as a hybrid. And that got us thinking that maybe that difference in plumage is only coded by a few genes, okay? Not many. Okay, so we wanted to know which genes are involved in the difference in plumage. And we went back to the field. All the lighter colors are localities that we had visited or other people had visited in the past. The darker ones are the new ones. And so what we did was go to many, many places, 29 different sites. This is difficult because this is Paramo. You have to walk a lot. They look very close, but they are not so much. And from those sites, we found one site where we could see a male of Chimborazo doing territorial displays and singing to a male of Jamesoni, which was one of the coolest days of my life. And then we did, with those samples, we did the genetics, the genomics, and we have here the three groups again, okay? There is a little bit of closeness between the central population and the southern population. That's what we see here in this cluster analysis around these individuals. We decided to trim our data set to get a data set that had very little missing data and get a structural analysis that was more trustable. These are the northern localities, a very good green block, right? We have the northern contact zone. These are not new specimens. This is what we had before. We had more Chimborazo specimens, and we're going to talk about this in a bit. We got the southern contact zone specimens, and then the southern ones, and then the very, very southern ones, which is really cool. Half of these individuals are males. They are very admixed, like, considerably admixed, but they all look like Chimborazo, okay? These ones are all males, and they look like Jamesoni. And one thing that I wasn't expecting is that how homogeneous this admixture is here. That, for me, you know more than me, it looks like as a population that was isolated for some time, okay, and by drift lost a lot of the other alleles, and then they got connected with this northern population and the southern population, and what we are really seeing is a pattern of hybridization by secondary contact. Leo Campaña, that is here in the room, is the responsible for doing the genomic analysis to try to find candidate genes for that plumage differentiation. And he was very disappointed that there are not many pigs. These birds look all the same in most of their genomes, and he tried different strategies to find those different genes, those very high FSTs, and he did find one gene that is very interesting. What you're seeing here is the summary of a region, so 15 variable positions in a bigger region, in a region that is close to BNC2, which is a gene that has been related to pigment saturation in human skin, which, melanin, structural colors could be playing a big role there, that relationship. These are the outgroups, okay? And they all have these, what we think are ancestral stages, okay? And then we have all these Jamesoni, and most of them are equally, like very similar, like identical to the outgroup. And then we have Chimborazo, and they have what seems to be a derived state. Okay, there are a few. The one Chimborazo that is a heterozygous. There is one Jensoni that has also heterozygosity. But I think we're in the right path. Maybe there is some epistasis going there with another gene that we don't know of. But I think we're doing things right. Leo is doing things right. What's next? Well, we have tested the phenotypic differences that are not there. We have to prove the obvious, that there is no an environmental gradient in our system. I think we have to do, prove it formally. And we have to do all the combinations of potential scenarios, right? We have to see the differences in gene flow during diversification or after diversification. We have to see the different splitting patterns, and we have to add the symmetric or asymmetric gene flow, which is one of the questions that is very interesting for us because we want to do female chose experiments. Our hypothesis is that females really prefer the green gorgeted males. Who wouldn't? I think that's just common sense. But we want to test that, and as we are speaking, we're trying to get a few more specimens to get them naturalized and combine the experiments with sound too. For that, I'm collaborating with Fernanda Duque, which is somebody that knows a lot about these high frequency vocalizations that the birds make when they are displaying. And we're going to do a lot of Sanger sequencing for those 15 variable positions in the BNC2 gene for the other individuals that we have in our sampling. This is hard, very hard field work, and there is a lot of people that I have to be grateful for their hard work. Paramo is not easy, and with that, thank you very much. If you have any questions or any ideas.
I was just wondering, do you have any idea about environmental variable? How do you going to integrate those things?
Well, we did already. We did some niche modeling, and I can show you. Yeah. So these are the paramours. These are the environmental, the niche models in the life Pleistocene. We have to separate them now. Those two sets, data sets, and compare them. But my impression is that they are going to be.
And have you found any association with these environmental variable with traits? No.
Nothing at all.
Okay.
These birds are almost the same. If you look them from afar, you wouldn't tell which one is which one because they behave the same, they eat the same, they're in the same environment.
Pretty
But
nice.
this is more like a formal thing that we have to do.
Cool. Thank you.
You're welcome. Yes. Sorry. Okay.
Thank you so much for the talk. It's a really, really interesting system. I was wondering, do you know what chromosome that gene that you presented is on?
Leo, do you remember which chromosome?
It's not on the Z chromosome.
It's not on the Z chromosome. I think it's not. But there was another talk in the conference where in hummingbirds they found the same.
Oh, that was actually what I was just about to say. Is there was a talk this morning where they implicated the same gene, I think, in coloration. So if you didn't know about that already, what do you do? So there you go.
No? No. Okay, I'll look for you. The next talk.
Good morning, everybody. My name is Lucas Mello, and I'm a master's student at the University of Campinas in Brazil. And today I'm talking a little bit with you about the diversification of neotropical tarantulas. When we think about the neotropics, we know that the neotropics is the most biodiverse region in the world. And for centuries, this pattern lots of scientists and naturalists think about hypotheses to explain this pattern, but we know that there isn't only a single hypothesis to explain it, and we are going to explore more drivers of diversification inside each of the domains of the neotropics to try to explain this pattern. So here we're going to focus on two humid forests of the neotropics, the Amazon and Atlantic rainforest, and the diagonal of open formations that is a diagonal that divides of seasonal environments that divide those forests, right? Which is composed by the Cerrado, which is savanna, and the Caatinga, which is a seasonal dry tropical forest. Well, talking about the drivers of diversification in the humid forests of the neotropics, one of the proposed drivers is the geomorphological processes in the Miocene related to the final uplift of the Andes, which altered lots of riverine flows in the Amazon, and quaternary climatic fluctuations, as well as riverine barriers, which led to the diversification of northern and southern Atlantic forests and connection between those two forests in the Pleistocene, in the quaternary, right? When you think about the drivers within the diagonal of open formations, we can point out two main drivers, which are the geomorphological processes in the Miocene in the Cerrado, related to the Central Brazilian Plateau, rearranging itself like to the form of plateaus and depressions that it has today, which is related to the diversification of lots of lineages of the Cerrado, which is pointed right here, and riverine barriers in the Caatinga, as for example, the San Francisco River, right? Well, talking a little bit, and it's configured, talking a little bit about tarantulas. They are the most species family in megalomorphs, with more than 1,100 species, and 1/3 of and 60% of this diversity is in the neotropics. So it's a lot of diversity, and we are trying to understand where it comes from. Well, the Dolichotherium genus is one of these genus. It is composed right now by 9 recognized species, right? Ranging from blackish, brownish, reddish spiders, and even metallic blue one that occurs in only one city. And when we think about tarantulas, we think about like bird-eating, plate-sized spiders, but these ones are more cute and pocket-sized ones. Like you can see in the photo. They live underneath rocks and logs in the dry diagonal, and in the diagonal of open formations, and they can live up to 8 years and have very long life cycles, right? The genus is distributed across all the environments that we talked already, and Campus Fictanos in Bolivia, right? Being an interesting model to study neotropical diversification. Well, our objectives, our aim are very straightforward. We want to describe diversification patterns in the genus and try to infer processes that might have been driving this diversification. Well, we went to the field to collect in the field and got samples from museums. We got 7 species of the 9 described once with 2 outgroups. We went for GBS, genotype by sequencing, to obtain SNPs to do our analysis, cleaned the data sets to remove linkage disequilibrium, and made maximum likelihood Bayesian phylogenetic inferences with datation, species delimitation analysis, and ancestral range reconstruction. Well, the first thing that I want to show is the maximum likelihood phylogenies. We did it in HAX, right? We can see that we have 2 monophyletic groups, one related to the Cerrado and one related to the humid forests, and 2 monophyletic groups, but not between them, from the Caatinga, right? We will keep the second tree here because the first one had Dolichotherium tucuriense, which is the Amazon species, but we had not a great sequencing of it, so we removed it from the analysis. when we look to the tree, let's focus on the branch of the Cerrado. Well, these blue lineages that we can see right here, we weren't expecting it to be blue. we were expecting it to be from Dolichotelli dominguense, which is the pink one, because they are morphologically very close and were used as dominguense in other works, and other revisions. And well, we wanted to understand if the blue lineages can be considered a new species. So we did species delimitation analysis with delimitear, with Build's demographic models, going from pamixi to independent lineages with gene flow, recent or ancestral, and tests the models with random forest classifier, right? Well, testing it, we see that two of the models that consider the species as the lineages as being independent are selected. We can't differentiate between them because they are very closely voted, but we can say that they are independent lineages. We did SNMF as well to see the genetic structuring of the lineages. And we got 5, 6, and 7 ancestral population as the best ones. Let's focus on the 7, k equals 7. And we see that they are 3 different lineages as well. So after we did this, we selected 2 individuals from every lineage that we had to do, another thing that we are considering to propose this as a new species is that it occurs in a region called Chapada dos Veadeiros, which is a formation with high levels of endemism in lizard, plants, and other groups, because it has something that's called campus rupestres, which are rupestran grasslands, physiognomy that is very, lots of species only occur there with high, it's a physiognomy with high levels of endemism. We can see it. We can further see that two of the other lineages are structured when we considered elevation, with the pink one happening in valleys and the green here, but the black one happening in the plateaus, right? Well, we chose 2 individuals from each of the lineages, and we did an analysis in SNAP, which is a method to do a species tree with Bayesian methodology, and we inputted a strict clock using a datation made with CO1 gene for the genus before. Well, we obtained 2 main times of diversification, one related to the Miocene and one related to the Pliocene. And with that tree, we made an ancestral range reconstruction, which the diva-like plus J model was selected. We see that in the first diversification period, we have the lineages occupying the great domains like Caatinga. Then it goes to the Cerrado. And from the Cerrado, it diversifies in the Cerrado lineage and in the humid forest lineage, which colonizes Caatinga later. And then in the second diversification time, we see diversification within the biomes. Well, we can see that in this first diversification time, it correlates with the expansion the beginning of the diagonal of open formations. The major lineages diversify in this period, and this period is also correlated with the increase of fire events and the under uplift, right? Well, in the second time, we see that the Caatinga lineages, which are the red one and the green one, remain stable, but this can be an artifact of the high cryptic diversity that tarantulas have. Like, tarantulas look alike a lot. And more studies in these lineages could reveal us other species that are there. Other pattern that we can see is that the Atlantic forest lineages divide itself in the northern and southern part of the Atlantic forests, but the diversification is a little bit before the Pleistocene fluctuations, so we believe that riverine barriers have impact in this pattern. And we see that the Cerrado lineages diversify within the Central Brazilian Plateau, as I showed before, in campos rupestres and plateaus and valleys, right? What it is expected to lots of lineages of the Cerrado. Well, we see that lots of lineages in the genus are related to rupestre and grasslands, so this might be one of the drivers of diversification inside tarantulas in the neotropics. Well, concluding, we see that studying diversification patterns in non-model groups is essential to understand diversification because we see other perspectives of diversification within this place. Geomorphologic, climatics, and river barriers can influence the diversification of neotropical tarantulas, and campus rupestres appear to be important to the diversification of this genus in the neotropics. Well, with that, I would like to thank my mentors, my co-authors, and my laboratory, as well as my funding agencies. And I know that this is a little bit off, but I would like to also incentivize you to consider being a blood and bone marrow donator because it can save lots of life and it's very important. Well, thank you very much.
Hi everyone, my name is Jenna McCullough. I just finished my PhD at the University of New Mexico, and I am starting a postdoc in the Zennel Ferguson lab at the University of Kentucky in a couple days, so. So the paradox of the great speciators describes a pattern in which some species are spread across many island systems and show little phenotypic differentiation across their range, whereas others inhabit the same ranges, but each island is phenotypically differentiated, and these were often considered subspecies in a larger species complex, polytypic species complex. When Diamond and Meyer coined this paradox originally, we didn't have a way to measure genetic distinctiveness of these great speciators, and therefore phenotypic differentiation was used as a general metric for genetic differentiation. And so the paradox is that because they occur across many island systems, obviously these lineages have a high dispersal ability, but since different islands have different differentiated forms, they were likely genetically isolated. And this would imply that they are poorly dispersing, and thus the paradox. So I like to think of these components of great speciators like a triangle with dispersal ability, genetic differentiation, and phenotypic differentiation as its three key components. And so for some context, this paradox was based off of geographic variation of Indo-Pacific birds, but not all groups of birds in the Indo-Pacific are considered great speciators. And they differ from other famous island-endemic adaptive radiations because great speciators involve rapid diversification without the evolution of obvious adaptive changes like bill morphology. And so one of the most prominent of the great speciators is the collared kingfisher complex, which was so important to Ernst Mayer's ideas that he put them right on the cover of his book, Birds of Northern Melanesia, in 2001. And so previous work on the systematics of Todiramphus kingfishers found that this great speciator comprised upwards of 10 different species and highlighted rapid diversification, but they did not find well-supported relationships within this complex or across the genus more broadly. And so one reason why could have been due to limited taxonomic sampling or character sampling. So for this project, for my dissertation, I wanted to look at the entire genus's geographic variation and look at fine-scale patterns of trait evolution to delve deep into the processes underlying a genus of great speciators. So theory predicts that island populations of a great speciator to be genetically and phenotypically differentiated and walk a fine line of intermediate dispersal ability. So for this project, we did moderate coverage whole genome resequencing of every single subspecies in the genus. So that's all the taxonomic diversity. So the genus has 29 species, but there are 93 phenotypically distinct subspecies comprised in multiple polytypic species complexes. From these data, we extracted 4 different types of molecular markers, and these data sets varied in total amount of data from 13 mitochondrial genes to 8,000 protein coding genes with 5 million amino acid sites. Across all data sets, we noted that the backbone of the Todiramphus tree is characterized by many short internodes suggesting rapid and successive diversification early in the history of this group. But instead of talking about the relationships, which I could do for days, some biogeographic themes in this tree include single island endemic species, continental species with no geographic variation or substantial geographic variation, geographic variation within a complex found on islands and continents, or only on islands. Moreover, there's variation in migratory behavior within species complexes. For example, one being migratory and the rest in the species complex being endemic to single islands. And finally, we have cases of sympatry on islands and also on continents. So altogether, it's complex, but they're not just geographically complex. I love this figure. My advisor hates it. So they're also phylogenetically complex. So we found a high degree of congruence between data sets and tree building methods with, for most of the species, with strong support, but there are some taxa bolded here that either showed different relationships or lacked support. Oops, what was that? Okay. So when comparing site and gene concordance factors across the two major topologies, we found that many of these short inner nodes were approaching values consistent with high incomplete lineage sorting. Moreover, we found cases of mitochondrial discordance between several subspecies of Pacific kingfisher, sasser, having some or all of the sequence individuals for these subspecies grouping with an entirely different species, sacred kingfisher, sanctus. And when we explicitly tested for gene flow between them, we found that the 5 mitochondrially discordant sasser taxa have elevated F4 ratios, suggesting, or with other sasser and sanctus, suggesting that these are hybrid lineages. And across the whole tree, we see signatures of ancient introgression ranging from 2 to 37% of the sampled genome. So overall, this helps to explain the topological incongruence between data sets and methods. So going back to our predictions, we see that gene flow is likely present as these lineages colonized islands. Now, what about the role of dispersal? Dispersal ability is at the heart of the great speciator paradox. Too much will cause pervasive gene flow, and too little will inhibit island colonization and speciation. Therefore, it's hypothesized that intermediate dispersers will have the highest speciation rates. However, when empirical phylogenies of great speciators have been used to test this hypothesis, they did not support this relationship. And so to test this here, we measured dispersal ability using the hand wing index, which is a measure of wing shape. Large values of this index describes more dispersive wings and vice versa for lower values. So to test the relationship between diversification rate and dispersal ability, we produced a time-calibrated tree using UCE data, secondary calibrations, and island ages in BEAST. We inferred a crown age of around 11 million years, and we found that most of the diversity has accrued in the last 3.5 million years. So here's an ancestral straight reconstruction of dispersal ability across the tree, with purples as least dispersive and yellows as the most dispersive. And we have box plots that show taxon averages. We see that the most dispersive lineages are those that are migratory or have established seasonal movements, which is expected, and that the least dispersive lineages include sedentary forest birds endemic to New Guinea and a clade of island endemic French Polynesian birds. So it's expected that sedentary forest birds would probably be less dispersive, but it's really paradoxical that taxa that have obviously dispersed to the furthest reaches in the Pacific also have the least dispersive wing morphologies. So when comparing estimated speciation rates and dispersal ability, we found that lineages with intermediate dispersal or intermediate values had the highest overall diversification rates compared to those with more extreme values. And though not perfect, this shows a similar bell curve as a simplified hypothesis that I discussed earlier. And the French Polynesian birds that I mentioned before are outliers to this pattern. So we tested some explanatory variables like island size and time since divergence, but they were not supported with phylogenetic linear models. However, I opted to leverage a famous pattern of declining land bird species richness in the Pacific. So each of these purple lines are eastern edges of different avian families. And we can use this pattern as a metric for an island's isolation based on the number of land bird species present, with islands very close to New Guinea having more species than those in French Polynesia. So on the y-axis, we have dispersal ability, and on the x-axis, we have increasing isolation. And what we see is that dispersal ability of island Totiramphus decreases with increasing isolation on the islands that they reside. So this has been shown before with skeletal measurements, but it's the first time that I know that this has been shown with wing shape. And overall, this declining pattern of hand-wing index and isolation suggests that wing shape is following the island syndrome. It means that predictable evolutionary trends are contributing to the erasure of the relationship between intermediate dispersal ability and speciation rate in this group. So returning to our predictions, we found that lineages with intermediate dispersal abilities do have higher rates of diversification, but that this relationship is being obscured by the island syndrome. Now, what about phenotypic differentiation? The defining characteristic of the great speciators is their incredible phenotypic diversity across island systems, and this has been focused predominantly on feather pattern and color. Hypotheses for why color might evolve rapidly include sexual selection, measured by the degree of sexual dichromatism, competition, in which sympatry drives divergent color patterns, and neutral processes, in which drift in small populations contributes to rapid evolution of color and pattern. So to study this, we collected a large spectral reflectance data set of representing all Totiramphus taxa across 15 plumage patches, and using these data, we see that the genus takes up 65% of all volume and color space. And we can use XYZ coordinates and chromatic and achromatic measures of color to do multivariate analyses of these two aspects of color for both sexes. Here, purple is slower rates and yellow is faster rates. And I'm showing male rates here, but we do see differences between the sexes. I didn't have time to talk about that today. And we found that males actually did not have the fastest rates of color evolution overall. And we can also see differences within species complexes, such as within Pacific kingfisher, sasser, and collared kingfisher, chloris. So, what is the relationship between the rate of color evolution and lineage diversification? The y-axis is color change, and the x-axis is lineage diversification rate. And we see that phylogenetic linear models indicate a positive relationship between the rate of color evolution and lineage diversification rate. This is true of both metrics that we use to calculate diversification rate, as well as for both chromatic and achromatic measures of color for both sexes. So in other words, more rapidly diversifying groups are evolving plumage color faster. And since the inception of the Great Speciator Paradox, this relationship has been assumed, but it's the first time, to my knowledge, that they have been empirically shown to be linked. So what is driving this rapid color evolution? Measures of whole body sexual dichromatism for both color and brightness were not supported as a predictor of rapid color evolution, nor did phylogenetic linear models support breeding or non-breeding sympatry as a predictor. To assess the role of drift, we use island size as a proxy for population size, and this too was not supported. But island size is just one proxy for population size. When we used heterozygosity estimates from the resequenced genomes, phylogenetic linear models supported a statistically significant and negative relationship between the rate of color evolution and heterozygosity. This was supported for both sexes and for both color rates. And these red outline dots here are estimates derived from topads and museum specimens, which we did have a significant amount due to the amount of taxonomic sampling for this project. And this is likely they're likely overinflated due to issues with degraded DNA. And so when I removed any of the taxa that were represented by topads, this pattern held with only half of the diversity still being represented. And so what this suggests is that smaller population sizes evolve color at faster rates in this group. And so this is suggesting that drift is contributing to rapid phenotypic evolution, which is really cool because this is a rarely invoked mechanism for phenotypic evolution, or for rapid phenotypic evolution. And so to wrap this up, we showed that the rapid pace of speciation has contributed to high ILS, but that these lineages are not completely genetically isolated owing to evidence of gene flow. and that the fastest diversifying lineages have intermediate values of dispersal ability, and that this pattern is being obscured by the island syndrome. And that faster diversifying lineages also have faster rates of phenotypic evolution, driven by drift in small island populations of this genus of great species eaters. And so I'd like to end by acknowledging that this work would completely not be possible without the countless collectors since the 1800s that have collected, preserved, and provided museum specimens for research like ours. The oldest specimen that we sequenced was from the 1860s from India, which was really exciting to be able to sequence. And so I'd also like to acknowledge our funding sources and the many people that have helped during my PhD. And I'd like to take some time, maybe like 1 minute, to do a question. So I didn't actually, I did a very large table looking at each patch and the color production mechanism, and I haven't technically looked at that, but a lot of these birds are structurally colored. And structural colors are known to evolve at faster rates. There's almost no carotenoid pigments. For the most part, none of these birds are red, but they do have a little bit of rufous and pheomelanin. But I think it is driven a lot by structural color. Yeah. Great. Thank you, everyone.